Hi friends and welcome to my channel Easy Life Science. Here we are talking about a structure of DNA and forms of DNA. The DNA is called as a deoxyribonucleic acid and it is a genetic material present in cells of plants, animals, algae, fungi, protozoa, bacteria and in some viruses. It is also present in a mitochondria and a chloroplast. In all organisms, the DNA is double stranded. However, in some viruses, it is single stranded. For example, 5x174. The double stranded circular DNA is present in E. coli. It is 1 millimeter in circumference and contains 4 into 10 to 6 base pairs. It has a molecular weight of 2.7 into 10 to 9. The structure of DNA is given by two scientists, James Watson and Francis Crick, in 1953. They proposed a double-stranded helical structure of a DNA, and for this structure, they got a Nobel Prize in 62, along with another scientist, that is Maurice Wilkins. The Maurice Wilkins proposed X-ray diffraction photograph of a DNA, and the structure of DNA, according to the Watson and Crick, shows some properties. The DNA consists of two polynucleotide chains or strands. The chains are twisted together in a clockwise manner and in a plectronemic manner to form a right-handed double helix having two grooves, one shallow groove, that is a minor groove, and one deep groove, that is a major groove. The strand runs in a opposite direction, that is anti-parallel direction. One strand runs in a 5' direction and other strand runs in a 3' direction. At 5' end, there is a phosphate group, and at a 3' OH end, at the 3' end there is a OH group. The strands are held together by a weak hydrogen bonds. Each strand completes a turn, and each turn has a 10 nucleotide base. Each turn occupies a distance of 30 angstrom and each base occupies a distance of 3.4 angstrom. The two strands are 20 angstrom wide. Each polynucleotide chain consists of a smaller subunits called as a deoxyribonucleotides. Each deoxyribonucleotide consists of three components. One is a base, sugar, and a phosphate. Base, sugar, and phosphate, these are components of one deoxyribonucleotide. Base, sugar, and a phosphate group. The base, there are two types of nitrogen bases, purines and pyrimidines. The purines are double ringed compound with a nitrogen atom at first, third, seventh and ninth position. Two purines are there, adenine and guanine. The pyrimidines are single ringed compound with a nitrogen atom at first and third position. Two pyrimidines of DNA are thymine and a cytosine. The purine and pyrimidine bases pair only in a certain combinations. Adenine pairs with a thymine and guanine pairs with a cytosine. The adenine pairs with the thymine with the help of two hydrogen bonds. The hydrogen bonds are formed between first and sixth position of adenine and thymine. Guanine pairs with a cytosine with the help of three hydrogen bonds. The hydrogen bonds in between them at a position first, second and sixth. So this is about the base. Next, about sugar. The sugar, that is a deoxyribose sugar. It is a pentose sugar having a 5 carbon atoms. 4 carbon atoms and a 1 oxygen atom forms a 5 membrane ring. The 5th carbon atom is present outside the ring. This is called as a deoxyribose because it lacks oxygen at a second carbon position. Here we can look it. At a carbon atom of 2 of a sugar, there is a lacking of oxygen, so it is a deoxy. But in case of RNA, there is a oxygen at a second carbon of a sugar. Then third component, that is a phosphate. The phosphate group is highly acidic, so the name for the molecule is called as a deoxyribonucleic acid. Each phosphate group is joined to a number third carbon atom of one deoxyribose and to the fifth carbon atom of another deoxyribose with the help of a phosphodiester bonds. So these are the three components of a one deoxyribonucleotides. Now the basic structures of the purines and pyrimidines. As said, the purines are double ringed compounds and pyrimidines are single ringed compounds. At position 1, 3rd and 7th, 9 in a purine there is a nitrogen atoms, and first and third bases of pyrimidine there is a nitrogen. Three pyrimides are there: cytosine, thymine and uracil. In DNA there is no any presence of uracil. It is present in a RNA, and in a RNA there is no thymine, there is a uracil. Cytosine, thymine and uracil, these three are pyrimidines. But out of these three, two pyrimidines take part in a formation of a strand, that is a cytosine and thymine. Then the purine, these are double ringed compounds. Example, adenine and guanine. Now some nomenclature of a nucleic acid component. Nomenclature for a nucleic acid component. Here the purines and pyrimidines are there. When a purine forms a bond with a deoxyribose sugar, it leads to a shine, or its name is always ends with a INE letter. For example, adenine, when binds with a sugar, it forms a adenosine in a RNA and a deoxyadenosine in a DNA. The some DNA inhibitors are there which can be used as a anti-cancer agent and anti-retroviral agents, because these inhibitors are nucleotide and a base analogous. At time of a DNA replication, they takes place some position of a particular nitrogen nucleotide base. So these are called the base analogous, and further the DNA replication stops. So they inhibit a synthesis of a DNA. Example, the anti-cancer agent like a 5-fluorouracil and a 6-mercaptopurine. The anti-retroviral agent which inhibit a DNA replication in a viruses or a retroviruses, for example, azidothymidine and dideoxyinosine. Now about the DNA structure. Here in this image there is a one polynucleotide chain, and this polynucleotide chain contains a number of deoxyribonucleotides. Here there is a 3-OH end, and 3-OH end means that the deoxyribose sugar, the carbon atom number 3rd, it is empty with a 3-OH, whereas the 5th carbon atom of the sugar is with the phosphate group. So these ends are called as a 5' end and 3' end. Towards the 3' end, OH group is there. Towards the 5' end, there is a phosphate group, and these numbers are numbers of a carbon atom of a deoxyribose sugar. And these phosphate groups or the two nucleotides are held together with the help of a bond, that is a phosphodiester bond. And whereas the nitrogen base are joined to the deoxyribose sugar with the help of beta N glycosyl linkage, and this linkage in between the position or 9th position of purine bases and the 3rd position of pyrimidine bases, which are joined to the one position of a deoxyribose sugar, or a first position of a deoxyribose sugar. And this is one strand, or this is one polynucleotide strand. Another strand is there, another complementary strand is there, and this another complementary strand is bound with this strand with the help of a hydrogen bonds. And traditionally the DNA sequence is down from a 5 prime to a 3 prime end. This strand can be written as a ACGTA. The second structure of DNA is a double helix. The second structure of DNA shows a two antiparallel polynucleotide chains which wound around a same axis. Sugar phosphate chain wrap around a periphery. The bases like adenine, thymine, cytosine, guanine occupy a core, forming a complementary base pairs in between adenine and thymine, guanine and cytosine on the rules given by Watson and Crick. The DNA is a double helix held together mainly by a hydrogen bonds. And hydrogen bond is a chemical bond in which a hydrogen atom of one molecule is attached to an electronegative atom, especially a nitrogen, oxygen or a fluorine atom, usually of another molecule. And as said, one polynucleotide chain joins or binds with another polynucleotide chain with the help of weak hydrogen bonds, and this bond in between a nitrogen base adenine pairs with a thymine and vice versa, guanine pairs with a cytosine and vice versa. There are two hydrogen bonds in between A and T pairs and three hydrogen bonds in between C and G pairs. The DNA has one ability or ability of a base stacking. The bases in a DNA are planar and have a tendency to stack. And these forces for stacking are a hydrophobic interaction and a van der Waals forces. Now about forms of a DNA. The crystallographic structure of a DNA shows three forms: A form DNA, B form DNA and Z form DNA. The B form DNA is a model which is proposed by Watson and Crick, and it has a property similar to that of the properties of Watson and Crick DNA model. And these are the property of these three DNA: A DNA, B DNA and Z DNA. The A DNA, it is a right-handed helical DNA. It has 11 number of base pairs in each turn. Rise per bases is 0.23 nanometers. It has a pitch of 2.46 nanometer. There is a presence of major groove. The Z DNA is called as a left-handed DNA. It has 12 number of base pairs in each turn. Rise per base along a axis 0.38 nanometers, and major groove is absent in Z DNA. And the characteristics of B DNA are similar to that of the characteristics of a Watson and Crick model. Normally, the B DNA, it has a one minor groove and a major groove. It is a right-handed structure. The base pairs are almost perpendicular to the helix, and these are 3.4 angstrom apart from each other. One turn of a helix is apart by a distance of 36 angstrom. Each turn consists of approximately 10 base pairs. The minor groove occupying a distance of 12 angstrom, whereas a major groove occupying a distance of 22 angstrom. In eukaryotic cell, DNA is folded into a chromatin. The double helical structure coils around a histone. The histone are basic proteins which are present in a nucleosome. DNA bonds to a histone and forms a nucleosome. The nucleosome is a fiber of a 10 nanometer. Nucleosome contains 146 number of nucleotides. The nucleosome contains a histone protein and a linker DNA. The histones are the basic made up of a basic amino acids and made up of units like H2A, H2B, H3 and H4, and they are in a octamer. This is a chromatin packaging a DNA. Further, the DNA forms a nucleosome or a structure like beads on a string. It gets wrapped around a histone and forms a 30 nanometer fiber, and such fibers forms a loop of 75,000 base pairs, and number of loops form a rosette like structure, and each rosette having a 6 loops. The number of rosettes forms a coil like structure. One coil has a 30 rosettes, and the two chromatids, 10 coils are there in each chromatid. This is a packaging of a chromatin. DNA melting and annealing. The double helical strand get denatured with the help of high temperature, and the two strands get separated, and this separation is called as a denaturation. At a high temperature more than a 95, the hydrogen bonds get breaked and the two strands get apart from each other. This is called as a denaturation, and this denatured strand further get associated to form a intact DNA, and this is called as a annealing, and for this there is a need of hydrogen bonds. So annealing and the denaturation. The temperature which is required for a denaturation is called as a Tm, and this temperature is depends upon a contents of G plus C base pairs, size of DNA, pH and ionic strength. The function of DNA. DNA consists of 4 bases adenine, guanine, cytosine, thymine that are held in a linear array of a phosphorodiester bond through a 3' and a 5' position of adjacent deoxyribonucleic moieties. DNA is organized into a two strands by a pairing of bases A to T and G to C on a complement strand. This strand form a double helix along a central axis. The 3 into 10 to 9 base pairs of a DNA in a human are organized in a haploid complement of 23 chromosomes. DNA provides a template for its own replication and thus maintenance of a genotype, and for a transcription of roughly 3,000 genes into a variety of messenger RNA molecules. So that's all. Thank you for watching my video. If you like my video, share it to your friends, and don't forget to subscribe my channel.